Thank you, everyone. We're going to start this session. We have an esteemed panel here with us today. Everybody come on and get seated. So this panel is regulatory pathway and applications in drug development for rare disease and case studies. So the purpose for this panel I alluded to yesterday. There's been a lot of recent approvals that, they disseminate into the broader community. There's, misconceptions sometimes about the totality of evidence that get discussed in the broader community. So what better way to address this was to assemble a group of people that could speak to different elements of, recent approvals. Obviously, there are some individuals that can't speak directly to their product, but they can talk through the steps on this panel. But what we wanted to do is address those so that people would get an opportunity to see, and hear examples of the dialogues and conversations, but also the levels of evidence that are needed to be done. So I'm going to start this panel with continuing the conversation we had regarding Reata, and Shannon, if you want to interject anything additional after you hear comments from the agency. We have Emily here, who actually did the review and can provide some background experience, and we'll come back and we'll interview people. And after this, I just want to start that for continuation, and we'll go down the row.
Sure, thanks. So for those that don't know me, I'm Emily Freilich. I'm the director of the Division of Neurology 1 at Cedar. And this is a good continuation since we just heard a lot about omaveloxolone and Reata. And I think Dr. Baracchio also mentioned yesterday some of the considerations that went into determination of substantial evidence of effectiveness for that application. As you heard, we have to determine substantial evidence, and typically it can come from two studies, a single study, or a single study plus confirmatory evidence. And as we also heard, Reata, omaveloxolone did have a positive, adequate, and well-controlled study that won on its primary pre-specified endpoint. But it had some limitations as well. It was a small study. It did not have positive findings on the secondary endpoints. And so it wasn't persuasive enough to stand alone as a single study. And so we really were considering what could serve as confirmatory evidence to support what appeared to be a positive treatment effect in the single pivotal study. And they had presented also some mechanistic data that showed that the drug seemed to be doing what it was purported to do. So some biologic plausibility that the drug was having its effect on NRF2 activation. And the natural history study, which we heard a lot about in the last session, but the benefits of that study, or the positives that allowed us to really rely on it, were what we heard a lot about was the natural history database, right? This FACOMS, where we had individual patient-level data and from a large number of patients that had systematically collected the same mFARS that was the endpoint in the primary, in the open label extension for the pivotal study. And so the other thing that was also compelling was that a lot of the centers were the same, and so we felt confident that the data was collected in a similar fashion. I think there was overlap on a lot of the centers between the natural history study and the open label extension study. And so looking at that data and seeing that you had several years of progression in the natural history data and could compare it to the 3 years of open label extension data on the same endpoint on the mFARS, and we're able to do the propensity matching because they had collected demographic data for baseline characteristics to be able to match on the important prognostic variables.
Thanks.
Hi everyone, my name is Lauren Boak. I work at the pharmaceutical company Roche. I've been there for 16 years. I've been very fortunate for the last 6 years to be working very closely with the HD community. I lead the tominersen program in HD, which is an example which I think a lot of people have, in the room have experienced. There's a couple of 3 steps forward, 2 steps back. rare disease drug development isn't easy, but we're all here to work together and see how we can make it a success for all the diseases, and really, really pleased to be here today.
Well, I think to start the session, I think just sort of segueing from Lauren's talk, your comment is, like, do you have any thoughts? I mean, as working in rare disease, obviously it's extremely different than big pipeline drugs. I mean, we all know the challenges, and I was wanting to see if each of you would convey just things that you felt there was a success in thinking, and then a whoopsie, we probably should have done it differently, as an example for your programs.
Maybe I can just kick us off because I started the segue. So we have been working in Huntington's for over nearly 13 years, actually, when we partnered with Ionis. But when we really at Roche started leading the development was about 7 years ago. And it was really important for us as we moved more directly into this space is to understand the disease, because we didn't really have that solid understanding. And what was really beneficial was the availability of real-world data. This is to understand the natural history. I mean, everyone in the room understands this, but even to understand what kind of clinical trials, what kind of endpoints we could expect to be able to use in our clinical trials. And we're very fortunate in HD to have access to a very rich set of data from the Enroll platform, which CHDI Foundation, we have representatives here, but also others in the HD community really came together to build. And I think everyone can agree that every single data point that is collected from a rare disease individual or their families is precious. And so the ability to be able to all have the access and ability to use all this data for all our purposes is so critical. I mean, this data set that we had really allowed us to hit the ground running with our clinical trial design and get started in HD. Obviously, there's still lots to learn and lots more to work together on. But I think that's just, having that foundation of data and information to sort of to start building, as we often referred to it, building the plane while we fly it. So that was really critical. But I'm sure there's lots of other experiences on the panel too.
Yeah, maybe happy to go next, as I've had an opportunity to work with CPAth and Colin and others for some time, and I often reference the fact that I would say most of my learnings, independent of formal education, has come from the School of Hard Knocks and learning what not to do. The rare disease development is very challenging, and this is my 27th year in industry, primarily focused on pediatric and rare disease, as I mentioned, and I noticed a pattern along the way with companies that I've worked for, that they tend to approach it in a fairly formulaic way, not too dissimilar from large pipeline products, which is a mistake. I think really trying to think differently about how you access patients, use of data sets, and I think regulatory science advancements and how we start to think about use of real-world evidence in our clinical programs, critically important. And from the last speakers from FDA, the reference to working with advocacy groups, in my mind, is a absolute critical success factor. If the advocacy groups don't understand at least the rationale, the fundamental rationale for your trial, good luck enrolling it. So just a couple of examples of really areas where I think are different than traditional drug development, large multi-center trials, and really encourage folks to really try to think differently as they approach rare disease development.
Joan, did you want to go next?
Yes. I would very much echo what you said about understanding the issues from advocacy organizations. And in Duchenne, one thing in particular that has been important is the advocacy organizations, PPMD in particular, stepping up with how do you sort out what the issues are in the endpoints that you're looking at. You know all of these things are important, but how do you look at even things like reproducibility and bringing companies, even before they're in the clinic, into that conversation in a non-competitive environment has been incredibly important. And CPAth is very similar in terms of bringing people together in a non-competitive, not just within the space, but beyond the space. So I think that is really important. I think one of the lessons that I have learned is the importance, as was mentioned earlier, of a biomarker to identify that your drug is on target, It is hitting the mechanism that you are looking at. It is supportive evidence in terms of mechanistic proof of concept, but it is incredibly important in terms of selecting a dose and because that is, I think, one of the Achilles heels in drug development in orphan, because we don't have the luxury of doing a 250 patient phase 2 study and choosing dose selection in multiples and then going on to a phase 3. We don't have the chance to do it over again. And that's in part because of the patients. It's also in part because many of the companies, yours excluded, are small companies that don't have a chance to keep doing it. So it's incredibly important to have that biomarker signal earlier, and that's been a lot of what we have also talked about in developing biomarkers with CPAP.
In terms of the positives and things that have worked, I think one of the things that has been great in this program has been the effort early on of translating the basic science and the mechanism of action into something that is functionally relevant. So there's been a lot of interaction with the patient community and with a lot of stakeholders and KOLs to learn that. And I think that's made the development successful up to this late stage. And I think that's again critical. And I think I'm echoing some of what the others have said. In terms of what could have been done differently, probably anticipating even more the complexity of a gene therapy in a rare disease and all the different things that you can't think of. And I don't know that anybody could have anticipated, just how complicated it gets in terms of safety and in terms of other aspects of development. Personally, since I come from a CNS space, I was used to gene therapy being delivered in the CNS, so it's much more comfy when you're hiding behind the blood-brain barrier than when you're outside of it. So, going back to your point about biomarkers and outcome measures, I think some of the environment that we just heard about now, in terms of the pilot programs and the FDA initiatives of developing outcome measures with sponsors, it would have been great to have this like 10 years ago when this program was starting.
So Curalis is a relatively young company, so we're learning from our mistakes, but more learning from the issues of others, right? But I'd really like to emphasize, which is where I think critical path really plays a role in ALS, is dogma. We have dogma handed down in ALS from the 1990s. The dogma includes and it's really around trial design. And every time a young company comes to me, they come with something that was published 10 years ago, and this must be the way to do phase 1, phase 2, phase 3. And there are 2 or 3 areas where the dogmas hurt us in ALS. And one of it is our primary outcome measure. If you read a paper about ALS, it talks about an ALS functional rating scale that was developed and published in the 1990s. Now, most of us were born in the 1990s, but Coco Gauff wasn't born in the 1990s, so that's how old the ALS-FRS is. And it was developed for a particular reason, to be able to document the point and the clinical point in the disease that the ALS patient that you're seeing in front of you was when you saw them. It was transformed through a situation in which the other primary outcome measure was survival. And you can imagine that survival is not a very comforting primary outcome measure for ALS patients, because it implies that at least half of them have to die before you get to a positive trial. So hence the ALS-FRS became the primary outcome measure, is the primary outcome measure, and was in the tofersen trial the primary outcome measure that failed the drug. I have a lot of opinions about that program. I'm a big recruiter, and I feel very strongly that the FDA made the right call. So you can get me going for an hour on that one. But buried in there is that the ALS-FRS was never built to be a primary outcome measure. It has become one They tacked on the respiratory component, but never validated it once the respiratory component was added. And then we implemented it across all clinical trials. What did we not do? We didn't look at how it was being given. We created these actually there's 3 certification programs for the ALS-FRS as a primary outcome in ALS clinical trials. One is American, one is European, one is Japanese. There's no surprise in this room, right? Yeah. These three programs, until last summer, all trained their raters differently. Yeah. So, and in their training, they certified, recertified only once every 2 years. So you have the situation where the regulators and the small companies who don't have anyone who is buried in ALS, seeing this as the gold standard, there's no flaws in it, it's a perfect outcome measure. This is where CPATH is going to help me a lot. It turns out that if you combine data from sites that are American, European, and Japanese, you're actually combining data that is not the same. It's called the ALS-FRS, but the raters score it differently. Yeah. If you change the rater, there's a huge internal bias. So you have an outcome measure that's determining the fate of drugs in a really important disease, in which you're combining data that has not been obtained in the same manner. So one of the big pushes last summer was that we harmonized the US and Europe so that ongoing trials will have one set of training for the ALS-FRS. We haven't yet brought in Japan. They use a different formula. And we haven't really addressed the issue of the fact that staff turnover at sites. Now, this sounds really far away from the FDA, right, and regulators. Mm-hmm. But they only have, the data that they look at is only as good as how we've obtained it. So there is a real need in ALS to take, and this is an example of a primary outcome measure. If I was a Parkinson's doc, I'd be saying the same thing. And if I was in FTD, I'd be even more extreme. But we have a real need to make this outcome measure operationally more robust. And so for us, in the ALS broader community, the partnership with CPATH is going to be critical. We've actually discovered that patients are better than raters and site staff. And we're actually really actively looking at self-administered ALS-FRS using an app, because you know what? It eliminates the bias of the raters and the site. So the only bias in the app is the patient's bias, which does not change over time. Whereas you change a rater, we had sites in the middle of a really important trial lose all of their staff over COVID, 100% turnover. So 100% of the patients had new raters. So you can tell I'm passionate about it. It's an example of how CPATH is going to help us. But the data quality should not be assumed, even if it's the primary outcome measure for 20 years. And we need to actually look at that. Mm-hmm.
Yeah. Tom or Lauren, do you want to talk to endpoints in your portfolio?
Sorry.
ALS is not an area
ALS, but,
that I'm particularly familiar with. But before you even got into the to the detail that you had referenced, immediately what was going through my head was thinking about the sensitivity, accuracy, and precision of a tool, and how reliable could it be if folks have to be trained on it. And so it makes perfect sense, the direction that you have. To the degree that it's possible, I very much, for various areas that I focus on, I very much like to get a comfort level before we even think about picking an endpoint to understand how reliable that endpoint is in an appropriately translatable model, in a non-clinical model. I don't know if that's possible in an ALS-like setting, but for many of the areas that I work in, it is. And I like to get comfortable with biomarker X, Y, or Z in advance of even proposing something like that to a health authority in a non-clinical model. But I prefer as quantitative as possible.
So I'm curious on this, if a potential solution would be switching to a functional milestone-based outcome, particularly in something that moves as fast as ALS, as opposed to continuing to try to improve the scale, which is less than ideal to begin with.
So that's an excellent part of the process, which is looking at, we're not at a point where we can move away from the ALS-FRS. We need to learn how to implement it operationally so the data quality is better across countries. But time to event, we're looking at different outcome measures that are much more common in oncology, say, but have a real relevance in ALS, it has real relevance in Huntington's, real relevance in FTD. So you can tell where else I've been. But the concept of pulling from more, not more established, but bigger areas like oncology, things like time to event or other outcome measures that you can more easily objectify and remove the operational aspects of the outcome measure.
The analog in Duchenne is the 6-minute walk.
Exactly.
12 years ago, the companies had different protocols for the 6-minute walk. So an assessor would use the wrong one. And so as opposed to now, there is basically a academic-based group that is training all the centers for all the companies. And it's a monopoly, but it's a good thing that everybody is getting the same training and getting it on a very consistent basis.
Maybe just I can add, it's obvious that there's so much that can be learned across diseases. And in Huntington's, as you mentioned, Angela, this sort of time to event proposal is something that is worth considering. And while we do have some endpoints that do okay in the later stages of HD, we can deal with them, we can do trials. Really the impetus is to move into earlier stages of HD in a similar way in Alzheimer's as well. And we're fortunate to have an integrated staging system that's both based on biological and clinical data. You'll hear more about that over the course of these couple of days. But this is really where we can learn about like time to a functional change, like from stage 2 to stage 3 in HD, or then looking at biological changes, because the clinical measures that we have do not change in HD from the earlier stages and up until a very, very late. And with our increased understanding of the underlying biology and how perhaps intervening earlier may be needed, this is going to be really critical.
So Colin, I'm wondering, I know this was on your list of topics to discuss, if this would help us solve another problem that you wanted to talk about, which is outcome measures to use for long-term follow-up or post-approval studies, right? Because you run into another issue of are these outcomes translatable to the real world? And a lot of what we do in trials, particularly outcome measures that require intense training and uniformity in how they're administered. So.
Just one additional comment to that is that obviously you're working in gene therapies. How are you attacking the follow-up periods and like standardization of what kind of data collection and following up patients who may move in and out of healthcare systems?
Yeah, it's a headache. I'm on that topic. I'm here to learn more than to convey, because we're struggling with this, obviously, and I think the input we get from KOLs and from patient communities is that, and I think I love your idea, right, of going to patient-reported outcomes, and I've learned over the years that in medicine, amazing things happen if you listen to the patients. So.
Tom, you have
Yep. While this concept as relates to regenerative medicines is new to us, it's not, the concept is not new, right? I mean, for pediatric rare disease studies, we have to do long-term follow-ups all the time. This is a typical thing. But one piece that's always fascinated me, and back to the headache topic, that I haven't been able to figure out yet is how to do better in the United States. I find that long-term follow-up is much more consistent, and patients are much more accessible outside of the United States. And I suspect it's because, and at least one of the drivers is we tend to change our medical homes a bit more here than folks in other countries. But this is a particular challenge that I would urge all of you that are in the development space to really think through strategically and tactically how you're going to keep track of these patients. It's hard.
Yeah, and it's particularly difficult in something like a gene therapy space where the administration of the therapy tends to happen in a big academic center that's all set up for this, but the follow-up won't.
Yep,
So
exactly.
very different instruments available sometimes.
So we have actually proof that it can be done. And it's not based on a drug development program, and it's not based in the US, so I apologize. But in Germany, the ALS Clinical Trial Network has done two different things that I think, in fact, I've connected people to really learn from. And one is that they have developed an app and put it out there to all of the German sites. And it's in a mobile phone ALS FRS-based app. And the patients in the clinics are using this app. I don't know how many data points, 10,000 data points or something they have over 10 years. And then the second thing they've done is they have set up a neurofilament assay program. And they now have several years, well, at least 2 years of data on all patients who are followed by the predominant ALS clinics in Germany. Their neurofilament levels every 6 months. So they have a huge data repository that they've built. But again, the issue of what country you're doing this in really comes up. Germany, people tend to stay in the clinics that they start with over time. Most of Europe is this way. The US is the opposite, even with a disease like ALS, particularly because what happens is people develop ALS wherever they're living. They often have to move to access their family resources in terms of care as the disease progresses. So even in a disease like this, you get a lot of movement across the US. Yeah. But I think there's still opportunities as we connect bigger groups to work on this. Mm-hmm. And I think this is going to be critical because we'd really like to get away from placebo-controlled trials in this disease. And we can only get away from it once our observational data is more robust.
Yeah. Okay. I mean, I know, Lauren, you spoke to this a bit earlier, and obviously CPATH is a big fan of data. How are you all using real-world data in your programs, and what things have you found useful for its use in various steps of rare disease drug development? Tom's a big fan, I know.
Yeah, I am. And we've had some recent success in this regard. We oftentimes have very challenging studies in pediatric settings. And in this particular example, this was a rare complication of prematurity. And it just so happens, and I tease my friends, I'll maintain anonymity of the other company, but they had done a very similar trial in the identical patient population about a year or so staged between us. And we looked at the control population from their data that they published in a very robust, high-impact, peer-reviewed journal. We spoke with our statistical colleagues internally about alternative statistical methodologies, in this case, a Bayesian-based approach. And we proposed this approach to multiple health authorities around the globe. And we effectively were able to borrow real-world evidence from a very well-conducted trial with, I'm sure, outstanding data quality, as is typical in our industry. And we're now registering this indication around the globe. So this is not going to be a solution for every trial that we pursue, and I guess we got a little bit lucky with having this cadence and pacing of the two studies. But what I also found, which was, I guess in retrospect, shouldn't have been a surprise, but when it was time to consent, the caretaker or mom and dad, our relative ratio of success was much higher than in our other studies. So our operational throughput for the study, the actual speed at which we could conduct the study, improved dramatically relative to traditional control methodologies. So again, this is not going to be a solution for any and everything, but in certain circumstances, I appreciate health authorities' openness to at least engage in the conversation. We still should be held to the same tier 1 level data standards that should accompany any trial, but in certain circumstances, this can work, and now we have some real proof that it can work.
Maybe I can add sort of more of the cautionary tale. Obviously, this is a wonderful example. And just maybe reiterating the importance of quantitative modelling and learning from as much data as possible. As mentioned earlier, that we have this wonderful registry of data in Huntington's on the Enroll platform, and which tracks the natural progression of disease. More in the later stages, there's still some gaps in the earlier stages of disease, but we can learn a lot from it. The phase 3 study from tominersen, and tominersen is an ASO targeting the Huntington, the underlying cause of HT. There was actually quite a lot of placebo effect, well, quote unquote placebo effect. And I mean, there's a lot of things that could have contributed to this. There was a lot of hope and expectation behind this trial. Then, it's a 2-year trial, so, perhaps placebo effect over time would start to diminish. But it's really just a reflection of how important it is to learn from not just natural history observational data, but also what happens in a clinical trial setting, and try to adjust for these factors. There may be factors around the ASOs. It's obviously intrathecally injected, and so there's some aspect of the actual modality and how it's administered that needs to be factored in versus an oral, etc. But all of these things are really important. Just reiterating the work that in the quantitative modelling groups at Critical Path do try to integrate, or even internal companies work on, to integrate these different factors. Because I think a few people have mentioned it, we'd love to be able to go away from placebo-controlled trials ultimately and be able to have these new ways of creating like digital twins, all of this technology that is becoming more and more available to us, but there still needs to be a foundation of data underpinning that. And so getting the right amount of data and the right amount of knowledge before we sort of embark on that is critical, and we can only do that when working together.
Anyone else have any thoughts?
So the ALS community has recognized one other data set that we need to figure out how to get access to, and that is the failed clinical trials. Mm-hmm. So there are trials that don't meet their primary endpoint due to trial design, but there are a number of clinical trials over the last decade in ALS where the trial simply failed. There's no difference in the lines, no matter what way you sliced it. That's an incredibly valuable data set. Mm-hmm. That data set that is essentially a full placebo data set, unfortunately. And some of these data sets actually have neurofilament data in them in ALS. And so you've got these big pools of, I can name 4 different relatively large trials for ALS. It doesn't compare to stroke, but it does. It's big for ALS, where that data could just help the entire community. And if we could if there could be a way to access it in a de-identified manner, where we could use that for our modeling, we could use that for our predictions, it would help us with a number of aspects of our clinical trial design. I think it's not real world evidence in the traditional sense, but it's a lot of evidence.
Yeah, we've done that in Duchenne as well. There are trials for. Yeah,
and just related to CPATH in particular, the consortia that I'm involved with, we do have a data lake amassing exercise underway as we speak. So this is an approach that is not unfamiliar to CPATH.
Yeah. Yeah. Can I just add one more thing? I'm sorry, I feel like I'm the cautionary tale telling. It's more just like a sort of a comment rather than, a question or a comment, like how, when we're looking at old data, particularly biomarker data from older studies, how are we sure and confident in the assay and the technical validation and the confidence in the data? I think that's a really important thing that we all need to be very mindful of. I think assays, NFL assays and other assays have really come made lots of progress now, but that's now. And so we just need to be to work together on how we can make sure that we can be confident. I think Dr. Baracchio actually mentioned it in one of her comments, that we need to start early to make sure that we do have that technical validation and confidence in the data, because garbage in, garbage out.
Indeed.
I can add to that a little bit of plug. I think RDCA-DAP is well suited to that, and I think that will be sort of on our agenda. But in addition to having the experts and the stakeholders at that and reviewing the data and trying to dissect it more thoroughly, with the QuantMed programs that we have, it's just a perfect marriage. One of the reasons I joined CPATH was just the ability to do these type of things.
Yeah, you had mentioned the placebo effect, and it's by aggregating this data. DRISC is looking to aggregate this data. CTAP has aggregated the data and looked at the question of whether or not the placebos are different than the natural history. And it turns out that they aren't in that disease. And it seems as though that is an effort-based measure, but at the same time, you see that the placebos, when matched, are very similar to, are basically identical to the natural history. So it goes to the how can we minimize the placebo in these trials. But knowing that information, that it doesn't differ from natural history, is critical to being able to go forward there.
And on the topic of natural history studies, this has been a new area of interest of mine in particular, because sometimes the real world evidence that you need or you might like to rely on simply doesn't exist. So sometimes the best real world evidence that can be created is what you create yourself. So we've been thinking about gating natural history studies for some of these rare disease programs for a number of reasons. Number one, to create the evidence that we could quality control and make sure that it's robust enough, unlike many registries, for example. But we're also getting, it's almost like an operational pre-feasibility, so you could get a sense of how the centers are actually able to access patients for you and who you can rely upon when it's time for the live game. So there's real value in thinking a bit differently about this type of approach and in these kind of circumstances.
I'll open up the floor to questions. I've hogged the question space, but if there's others that want to ask questions, I see Susie McCune getting up. I'll let her, others.
Hello, I'm Ahmed Pindon, Chief Scientific Officer of VCP disease. We are a particular disease because we have one mechanistic, the VCP, but the problem is that we have patients in ALS, in Charcot-Marie-Tooth, in myopathy, Parkinson's disease. So on the first part, preclinical, we have one target, one mechanisms. But then after, when it comes to outcome measures, to how can we do that and integrate all these different manifestations, how can we, in drug development, think about outcome measures that can be across all those diseases. And I think we are not the only disease with this problem. And if you take the rare disease from ALS point of view, for example, you have to choose which one of all the genes you are going to choose. So if you take the other approach of centering on a mechanism, but then after, I think it's a headache for all of us, how after that, in clinical phase, how can you make the outcome measures?
So you talk about phenotypic divergence from the mechanism, right? And how you
single mechanism, but then you have from ALS to Parkinson's to Charcot-Marie-Tooth.
And there are no biomarkers that reflect pharmacologic engagement downstream that would be common to all these phenotypes?
Well, that's what we are looking for. We are looking into NFL, we are looking in this kind, but on the really measuring the outcomes, that's our headache, and I think it's something we should think about because we approach rare disease by ALS or Charcot-Marie-Tooth, but in fact it's not one disease, it's a lot of disease.
There are attempts now, right? And the Wave Life Sciences did a basket trial with their C9 targeted therapy. So there are natural partners and disease partners for basket trials. Some are very, very different, and NFL was a good comment because, but they successfully ran a study with ALS and FTD in the same SAD-MAD study. The study is closed, but the ability to run, they demonstrated you could do that. But each of the arms had the outcome measures appropriate for that disease. What was common was the biomarkers. And so I think that's in situations where you have a molecule that is targeting a pathway that is common across, say, neurodegeneration, I think that's a very doable thing as we get more robust biomarkers. And that's going to be the essence. And then once you get through your SAD-MAD into POC and you're looking at your pivotal, that's when you split off. But your early data will be driven by biomarkers, in my opinion.
I like that.
Thank you.
Susie? You're like...
Susie goes first.
Age before beauty, right?
Wisdom.
I'm Susie McCune. I'm at the clinical research organization called PPD. And just wanted to remind everyone, I'm in the pediatric space with Tom, and the Duchenne space as well. About 50%, if not a little bit more, of rare diseases are pediatric. A lot of them are congenital. And so we see manifestations from infancy through childhood, through adolescence, or toddler, childhood, adolescence. And the outcome measures change over the course of each of those periods. The way we capture them change, especially quality of life questionnaires, especially for the patients and the caregivers. And as they get into adolescence, their approach to things may be very different from their parents. And so the 6-minute walk test being one that reminded me not only of not being able to do it when you're really young, but also I heard stories of parents literally grabbing their child's hand and dragging them down, the runway. Right, so just a reminder that that's a complication in pediatrics as well, as we struggle with outcome measures, period, but then add on to it that they will differ through the course of the life of a pediatric patient.
Yeah, and we do, and I suspect many of my colleagues that do pediatric studies often strive to go as low in the age range as benefit risk will support. So, but this is the inevitable challenge that we have, right? What measure of exertion is appropriate for an infant versus a child versus a teenager?
And even the quality of life questionnaires, have they been validated?
Exactly.
Yeah, and
Preaching to the choir, guys. Well, anytime I can say neonates, and certainly with congenital rare diseases, we do see the manifestations. Many of them, especially the severe neurodevelopmental manifestations in patients in the NICU that can't get off the ventilator, and so looking at outcome measures for those patients that aren't just survival, as was mentioned earlier, but also functional outcomes.
Thank you, Susie. Good to see you.
Yeah, so Klaus Romer with CPATH. I just want to introduce one clarification and then a question. Time to event is really a data analytics approach that can be used to define the primary analysis of a trial, not necessarily an endpoint per se. But Angela, thank you for bringing up the fact that we need to stop what a friend of mine calls the last paper carried forward approach.
Yes.
We definitely need to stop that. But thinking about the longitudinal component, how important it is, that's one of the beauties of the time to event, time varying probability and all that, looking at the entire movie throughout the trials. Speaking about that, can the panel comment a little bit on you guys' take on the use of digital health technologies in these kinds of trials for rare diseases?
Who wants to start?
I can start.
We all have an opinion.
Yeah, I think digital health technologies are very, very promising, and because clearly the amount of data and that you can collect outside of clinical visits it provides more sensitive evaluation of effects over time. I think we still need to be very conscious of the additional burden this takes in clinical trials as well, and it's always about making sure that everything that you collect is going to be useful and used used for a purpose, because it's time and burden on individuals and their families during the clinical trials. But overall, our experience, we've got experience in Duchenne's, but also in Huntington's and Parkinson's of developing digital monitoring tools, and it's really at this point in time very much complementary to what's existing. But I'd love to see a future where these will be the tools that are only needed, because they particularly may be more sensitive early on in stages of diseases. And yeah, it's just adding to the tool armatorium that we need for our drug development programs.
It's kind of natural in ALS, because ALS is a movement-based disorder, right? You lose various movements, so it is definitely, there's a lot of interest. The simpler, the better. Digital health, they like really complex things, right? And that gives you a lot of data. And I like simpler things because I want cleaner data. But I think it's a huge step forward. And it actually applies to a number of neurodegenerative diseases, Huntington's, ALS, but PSP, certain variants of FTD. It's actually going to be a huge tool.
There's actually been a very successful track record of developing it in Parkinson's, and it's moving in that direction a lot. So I think it's the future. And one tidbit that I've learned actually trying to develop an app for Parkinson's is that adaptation, the take on of technology and the use and the compliance with it vary a lot, and it tends to go up if the patients get some benefit or some use out of it, as opposed to just being a chore and wearing it so the doctor is happy. So we tried to develop an app for Parkinson's. Nobody was using it, and when we made it dual function to remind patients to take their medications, then everybody was using it. So I think it's something to keep in mind.
The same in Duchenne, that if you're looking at a muscle disease and you're looking at function, you're looking at how fast the kids run every day and what their maximum speed is, or they're close to maximum speed, that's clearly speaking to feel and function. Absolutely. And you see it in it's real world evidence, as opposed to the 5 seconds to go upstairs in a clinical trial setting.
And then I think this is especially true in studies or in patient populations that have a pediatric component as well, because if you could measure something without requiring an office visit or another stop, you have to remember it's not just the patient in your trial, it's the caregivers that have to get them there, it's missed work, missed school, missed afternoon sports, so forth and so on. So trying to measure as much as we can reliably and robustly without requiring a car trip or a plane trip, I think benefits everyone.
I just wanted to make a comment. We seem to have had a really lengthy conversation on endpoints, and I think it's important to remember a couple things with some of the questions that have come up. So first and foremost is, what is that patient experience with that disease? Because even in families of diseases or genetic pieces that which disease will appear, the example with FTD and ALS, those patients' individual experience is going to be very different, and what they really think about their outcomes and their progression of their disease is going to be very different. And so I think we need to not forget about that when we're thinking about the family concepts or the mechanistic concepts, is that in the end, how a patient feels about their disease and their experience is going to be very unique and needs to be critically considered when selecting an optimal outcome to support a clinical trial endpoint. So I think we need to remember that, because while mechanistically it looks great, it may not actually be the right thing to measure in the end, which ties to the other pieces. What do we think our drug's going to treat or our biologic's going to treat? And that's the piece that I think a lot of folks sometimes forget about when they're selecting outcomes and endpoints, right? We're really good at taking in the patient voice, and we've come a long way in that way. But we also have to make sure we know that what we think the treatment's going to do, we're measuring that. So we're not questioning when we get the data that comes in and reviewing it, going, well, how far off was I in what I captured? And so that's why we encourage early and often engagement with the agency. CDER, CBER, come talk to us. We want to help optimize those trial protocols and endpoint selection when possible. But we can only do that unless you come talk to us. And so I think that's a really important conversation when we're talking about our rare disease clinical trial development because we don't have a lot of opportunities. And we definitely are on the same side of we don't want to waste patients. We don't want to waste, we only have a small amount of patients to be able to recruit from. So I just want to remind everyone to do come and talk to us early and often.
Last question,
Great,
Susan.
thank you.
Susan Ward, CTAP. I thought this was a wonderful discussion of endpoints and all the pitfalls and ways of trying to avoid them. And ditto around use of real-world data. But there's one piece that I think I'd really like to hear people speak to that hasn't been raised, and that is accounting for heterogeneity. And, you know, we talk about heterogeneity a lot, and sometimes that's different symptoms for different patients. But I think Duchenne is an interesting example because for each individual patient, the journey is remarkably consistent. What's different between patients, I mean, in the scheme of things, and patients may not say that, but from an observational clinical perspective, there's tremendous consistency, except for the rate at which the disease progresses. And so the heterogeneity is not so much around the symptoms, it's around the rate at which patients move through it. And so the question I want to raise is that there are ways in which one can, I mean, obviously that leads to a lot of variance in trials. And obviously you want to overcome that, but you can have perfection in all your endpoints. You're not going to get away from the fact that that disease is moving at different rates in different patients. So to what extent do you guys rely upon or utilize even statistical approaches that you can use in analysis, not design, but in analysis to reduce the variance?
So I'll make a quick comment and then hand it down because I think others have more. In ALS, we have a problem with statistical analysis.
Yeah.
There are at least 12 different statistical approaches to the ALS-FRS. So one of my requests that Colin and Elsa are receiving are actually to bring, there are brilliant statisticians that have been involved in ALS trials over 20 years, and yet there's still 12 different ways of analyzing it. And then when the company puts it to the FDA has to reanalyze it because they're all using different. So one of the things that we have to do is make the statistical analysis plans more homogeneous, right? And then within those plans, we need to stratify and follow. We do have some, in ALS, people talk about heterogeneity, but it is a disease of motor neuron loss. So there is a course, and people follow that course, and they're either fast, normal, or slow progressors, even though the milestones may look different along the way. So we now have a number of algorithms that have been developed that can predict whether or not it's a fast, normal, or slow progressor. So that's starting to be incorporated, but it's at the beginning.
And I think the dilemma that we have as it relates to prioritizing a purely statistical strategy is the sample size that you're starting with in the first place. These tend to be relatively modest sample size studies, particularly in the ultra-rare diseases, right? So your ability to meaningfully stratify based upon a statistical strategy alone is going to be tough. I prefer more of an operational approach. The concept of basket trial came up earlier. If we're able to endotype the disease in early development, if we know that there are different courses of disease progression, for example, and then test your treatment effect early on, if there is one, and then prioritize that patient population for your subsequent late development programs. So I prefer more of an operational approach to homogenize than a statistical one.
Yeah, I mean, what we've done in Duchenne is a bit of an all of the above, and Susan, I don't think you can decouple the analysis from the design. I think you need to go in with keeping that in mind. And for anyone who doesn't know, so Duchenne, everybody follows this course where they improve just by natural development, and then around age 7 they start going down. And there's very few predictors. There are some predictors that can maybe suggest that the kid is going to go faster or slower, but they're not great. So I don't know, other than following the advice from one of my mentors of making friends with a good statistician, I don't have a solution.
Yeah. I would say, though, even though they're not great, they improve things.
Yeah.
And so just
Sure.
if you can improve the signal to noise,
Right.
you can improve the power consistently. So by adjusting for baseline function, as well as potentially as we learn more, these are long trials, so we learn more, we may end up analyzing it with additional information about what study population within that group. And you may not have to define it operationally, but you may be able to do your primary endpoint on a smaller group that's more homogeneous with additional information.
Yep, great.
I'm just building on what everyone has already said, really. It's the enrichment at the beginning of the study is really important, plus also the pre-specified analyses of subgroups. And I mean, Huntington's similar to Duchenne's. We understand the underlying cause, but there's still a lot of variability. There's a mutation, but then there's different CAG repeats, and the number of CAG repeats only accounts for 50% of the variability. And so the worst possible thing is that you do try to study and then it fails because you haven't predicted your placebo decline appropriately. And so trying to understand what factors that you can adjust for at the beginning, algorithms, as you mentioned, Angela, we have prognostic indexes that are integrating different data points. We can only get better though, because the more that we can at least understand what the trajectory, if we're using placebo arms, or to design our trials at the beginning, because as I mentioned already, the last thing you want to do is it to be a clinical trial design error, the reason a trial fails. It needs to be, no trial is a failure. Obviously, we learn a lot, even if it doesn't meet what we were expecting. But we can try and do as much up front, but also at the statistical analysis plan stage as well.
Well, thank you, everyone. We'll close this session. We appreciate the expertise of this group.